Welcome to today's lecture on polymerase chain reaction, commonly abbreviated as PCR. This technique is a cornerstone in molecular biology and biotechnology. Today we'll dive deep into how PCR works, its significance, and how it applies in various domains such as medicine, forensics, and biological research. Polymerase chain reaction was developed by Dr. Kary Mullis in 1983. His revolutionary idea allowed scientists to amplify specific DNA sequences rapidly, easily, and with high specificity. So significant was this innovation that Mullis received the Nobel Prize in Chemistry in 1993. Before PCR, amplifying DNA was a tedious and time-consuming process. Now, even minute amounts of DNA can be exponentially amplified, opening up new frontiers in genetic analysis, diagnostics, and evolutionary biology. In fact, if you think about it, PCR is akin to a molecular photocopier, making millions of copies of a DNA fragment in just a few hours. Throughout this lecture, we'll cover the underlying principles of PCR, the steps involved, different types of PCR, and its real-world applications. I will also emphasize the key points that are frequently tested in exams like NEET and other entrance assessments. So, whether you're aiming to become a researcher, a medical professional, or simply want to master molecular biology, understanding PCR is essential. Let's begin by exploring the fundamental concept and principle behind this technique in the next slide. Let's now delve into the principle of PCR. At its core, PCR is based on the natural process of DNA replication, but with a twist. It's performed in a test tube, or more precisely, a reaction tube. The objective is to amplify, or make multiple copies of a specific DNA segment. PCR does this by mimicking the cell's DNA replication machinery, but in a controlled laboratory environment. What sets PCR apart is its reliance on thermal cycling, alternating between different temperatures to carry out three major steps: denaturation, annealing, and extension. The key to PCR's success lies in its reagents. 1. Template DNA. This is the DNA containing the region you wish to amplify. It could be from blood, a tissue sample, or even a single hair follicle. 2. Primers. Short, single-stranded DNA sequences that are designed to be complementary to the target region. They serve as starting points for DNA synthesis. 3. DNA polymerase, an enzyme that adds nucleotides to build the new DNA strand. The most commonly used is Taq polymerase, which is heat-stable and was isolated from the thermophilic bacterium Thermus aquaticus. 4. DNTPs. These are the building blocks: deoxynucleoside triphosphates, A, T, G, and C. 5. Buffer system. Maintains the optimal pH and provides necessary ions like Mg2 for enzyme activity. During each PCR cycle, the DNA is first denatured by heating to separate its strands. Next, the temperature is lowered to allow primers to bind, or anneal, to their complementary sequences. Finally, the temperature is raised slightly so the DNA polymerase can extend the primers and synthesize new DNA strands. This cycle is repeated multiple times, typically 25 to 35 cycles, leading to exponential amplification of the target sequence. Understanding this principle is crucial for interpreting results, troubleshooting experiments, and applying PCR in practical scenarios. Now, let's look more closely at each component that makes PCR possible in our next slide. To carry out a successful PCR, understanding each component of the reaction is crucial. Let's examine these elements in detail. The PCR mixture typically includes 5 core components: template DNA, primers, Taq DNA polymerase, DNTPs, and a buffer system, usually supplemented with magnesium chloride. First, template DNA is the strand that contains the sequence you aim to amplify. This could be extracted from a variety of sources: blood, tissue, plant material, or even ancient bones. The quality and purity of this DNA influence how well PCR will work. Even minute quantities of template, nanogram or picogram levels, can suffice due to the sensitivity of PCR. Next, we have primers. These are short sequences of nucleotides, usually 18 to 24 bases long, designed to flank the target DNA sequence. Primers are custom-made and are critical because they define where the polymerase starts copying. A poorly designed primer can result in nonspecific binding or no amplification at all. Third, Taq polymerase is the enzyme that synthesizes the new DNA strand. Isolated from the thermophilic bacterium Thermus aquaticus, this enzyme remains stable at high temperatures used in PCR around 95 degrees Celsius. This thermostability is what makes Taq polymerase ideal for PCR, where repeated heating would denature ordinary DNA polymerases. Then we have DNTPs, or deoxynucleotide triphosphates. These are the building blocks of DNA: adenine A, guanine G, cytosine C, and thymine T. They are incorporated into the growing DNA strand during the extension phase. Finally, the buffer system maintains a stable pH and provides the necessary ions for enzyme activity. Most importantly, magnesium ions, often supplied as MgCl2, act as a cofactor for Taq polymerase. Too little magnesium, and the enzyme won't function well. Too much, and you may get nonspecific amplification. Altogether, these components are combined in a precise ratio to form the PCR mix. This solution is placed in a thermal cycler, and the magic begins. Now that we've covered the components, let's look at the 3 critical steps in the PCR process: denaturation, annealing, and extension. These are repeated in cycles, typically 25 to 35 times, depending on the desired yield. Denaturation at 94 to 98 degrees Celsius. This is the first step of each cycle. The double-stranded DNA melts open to single strands, allowing the primers to access their target sequences. The high temperature breaks the hydrogen bonds between base pairs, essentially unzipping the DNA. It usually lasts 20 to 30 seconds. Annealing at 50 to 65 degrees Celsius. The reaction is cooled to allow primers to bind or anneal to their complementary sequences on the single-stranded DNA. The precise temperature depends on the primer's melting temperature, Tm, which is determined by its sequence and length. If the temperature is too low, primers might bind nonspecifically; too high, and they may not bind at all. Extension at 72 degrees Celsius. This is the optimal temperature for Taq polymerase to function. The polymerase starts adding DNTPs to the 3-foot end of each primer, extending the DNA strand and synthesizing a new double-stranded molecule. The duration of this step depends on the length of the DNA fragment being amplified, typically 1 minute per kilobase. These steps form one cycle. Since each cycle doubles the amount of DNA, the amplification is exponential. After 30 cycles, more than 1 billion copies of the original DNA segment can be produced. Think of it like a chain reaction, hence the name polymerase chain reaction. In practice, a preliminary denaturation step is often added at the beginning to ensure complete separation of DNA strands. Likewise, a final extension phase is included at the end of all cycles to ensure that any partially extended products are fully synthesized. In the next slide, we'll explore how these cycles are timed and how the thermal cycler manages this precisely. The success of PCR doesn't only depend on what's in the reaction tube, but also on how precisely the thermal cycler changes the temperature during each cycle. PCR is a time-sensitive and temperature-sensitive technique, and here's how the typical thermal program looks. We begin with an initial denaturation phase, typically at 94 to 98 degrees Celsius for 2 to 5 minutes. This ensures that all DNA strands are fully separated before cycling begins. Each PCR cycle then consists of denaturation, 94 degrees Celsius for about 30 seconds. This melts the DNA strands. Annealing, usually 50 to 65 degrees Celsius for 30 to 60 seconds. The precise temperature depends on the primer's Tm. Extension, 72 degrees Celsius for around 1 minute per kilobase. If your target is 500 BP, a 30-second extension might suffice. For 3 kb, at least 3 minutes is needed. Most protocols run 25 to 35 cycles. The more cycles, the more product you get, but more is not always better. Too many cycles can lead to non-specific products or depletion of reaction components. After the last cycle, a final extension step at 72 degrees Celsius for 5 to 10 minutes ensures all DNA strands are completely extended. Finally, The sample is held at 4 degrees Celsius to stabilize the product until it can be analyzed or stored. Thermal cyclers are programmed to execute these steps automatically, switching between temperatures rapidly using metal blocks and Peltier elements. The precision of these temperature transitions is essential to PCR success. Up next, we'll look at the different variations of PCR that have evolved from this basic protocol. Over time, the basic PCR technique has evolved into several variations, each tailored to specific applications and improving upon limitations of the original method. These modified forms of PCR increase sensitivity, specificity, and versatility. Conventional PCR. This is the classic method we've discussed so far. It's used for simply amplifying a target DNA sequence. The final product is usually analyzed via agarose gel electrophoresis to confirm size and yield. Real-time PCR. qPCR. This advanced technique allows you to monitor the amplification of DNA in real time using fluorescent dyes or probes. This means we don't need to run a gel to see if PCR worked. We can watch DNA amplify as the reaction happens. The fluorescence correlates with the amount of DNA, allowing quantification. It's widely used in diagnostics, including viral load testing like HIV and SARS-CoV-2. Reverse transcription PCR. RT-PCR, not to be confused with real-time PCR. RT-PCR starts with RNA, not DNA. An enzyme called reverse transcriptase is used to convert RNA into complementary DNA, cDNA, which is then amplified. This is crucial in studying gene expression and detecting RNA viruses. Nested PCR. This involves 2 successive PCR runs. The first set of primers amplifies a broader region. A second set of primers, nested within the first, is used in a second round. This increases specificity and is useful when the DNA template is low or contaminated. Multiplex PCR. This version uses multiple primer pairs in a single PCR reaction to amplify several targets simultaneously. It's efficient, cost-effective, and commonly used in pathogen detection panels or genetic screening. Each of these techniques has a place in modern biology and medicine. For NEET aspirants, it's essential to distinguish between them and know their applications. For instance, qPCR equals quantification, RT-PCR equals RNA, nested PCR equals high specificity, multiplex equals multiple genes. PCR isn't just a lab trick, it's a powerhouse tool with applications that span many fields, from clinical diagnostics to criminal investigations to evolutionary biology. Let's take a closer look. Medical diagnostics. PCR is widely used to detect genetic disorders and infectious diseases. For instance, it can detect specific mutations in the BRCA gene linked to breast cancer or the BCR-ABL fusion gene in chronic myeloid leukemia. During the COVID-19 pandemic, RT-PCR became a household term. It was the gold standard for detecting SARS-CoV-2 RNA. Forensic science. In forensic investigations, DNA from crime scenes, often degraded or in trace amounts, can be amplified using PCR. Short tandem repeat (STR) analysis uses PCR to create DNA fingerprints. This is how suspects are matched to evidence. Genetic engineering and molecular cloning. PCR is used to clone genes, tag them, or insert them into plasmids. It's essential for recombinant DNA technology. You can amplify a gene from one organism and introduce it into another for expression studies. Research. Whether it's checking for gene expression, creating genetic knockouts, or mapping genomes, PCR is at the center of modern molecular research. Real-time PCR is used extensively in gene expression studies with quantification of mRNA levels after reverse transcription. Agriculture and food safety. PCR is used to detect genetically modified organisms, GMOs, in food products. It's also used to detect pathogens in food and water, like E. coli or Salmonella, making it a key tool in public health. Environmental biology. PCR helps identify microbial communities in soil and water, track biodiversity, and even study ancient DNA. As you can see, PCR's versatility makes it one of the most important techniques in biology today. In the next slide, we'll explore the limitations and challenges associated with this powerful tool. While PCR is incredibly powerful, it's not without its challenges and limitations. Understanding these is crucial, especially when interpreting results or troubleshooting an experiment. Contamination. One of the biggest issues in PCR is contamination. Because PCR is so sensitive, even tiny amounts of foreign DNA, like your own skin cells or DNA from a previous reaction, can be amplified, leading to false positives. That's why labs use separate areas and sterile techniques for PCR setup. Primer design. The success of PCR depends heavily on the primers. If primers are not specific enough, they may bind to unintended sites, leading to non-specific products or primer dimers. Designing primers requires knowledge of the target sequence, melting temperatures, GC content, and absence of secondary structures. Lack of proofreading in Taq polymerase. Taq lacks 3 to 5 exonuclease activity, meaning it doesn't correct mistakes. This can introduce errors into the amplified DNA. For applications like cloning or sequencing, high-fidelity polymerases with proofreading abilities are preferred. Inhibitors in the sample. Biological samples often contain substances that inhibit PCR, like hemoglobin, heparin, or polysaccharides. These can interfere with polymerase activity and cause amplification failure. Quantitative limitation in conventional PCR, regular PCR, is endpoint analysis. It doesn't tell you how much DNA was there originally. That's where real-time PCR shines, providing quantitative data. Size limitations. Amplifying very large DNA fragments, 5 kb, is challenging with standard PCR and may require special enzymes and conditions. In the field or during NEET exams, understanding these limitations helps in interpreting both practical outcomes and theoretical questions. We'll now look at some high-yield NEET-specific information you must commit to memory. This slide is tailored for those of you preparing for NEET and similar entrance exams. Here are some high-yield facts you're likely to encounter. Kary Mullis. Remember this name. He invented PCR in 1983 and received the Nobel Prize in Chemistry in 1993. Taq polymerase, sourced from Thermus aquaticus, a bacterium that thrives in hot springs. It is thermostable and withstands the high temperatures needed for denaturation. PCR steps. Know the temperatures by heart. Denaturation occurs at around 94 degrees Celsius, annealing at 50 to 65 degrees Celsius, and extension at 72 degrees Celsius. These may appear as direct match the following or MCQ questions. RT-PCR, often asked in the context of RNA virus detection. COVID-19 diagnostic tests used RT-PCR extensively, which first converts RNA to DNA before amplification. Real-time PCR, qPCR, don't confuse with RT-PCR. It's quantitative and uses fluorescent markers. Important in diagnosing cancer gene expression or measuring viral loads. Primer design. NEET questions sometimes ask what is essential for primer design. The answer is specificity to target sequence, optimal melting temperature, and GC content around 40 to 60%. dNTPs act as substrates for the polymerase. A question might ask, what provides nucleotides during PCR? And the correct answer would be dNTPs, not ADP or DNA ligase. Mg superscript 2 acts as a cofactor for Taq polymerase. If missing or in wrong concentration, PCR won't work. Revising these facts regularly and practicing related MCQs can give you an edge in competitive exams. Real-time PCR, also known as quantitative PCR, qPCR, is a powerful upgrade to conventional PCR, enabling both amplification and real-time monitoring of DNA during the PCR process. Widely used in diagnostics, especially during the COVID-19 pandemic, qPCR is highly sensitive, accurate, and rapid. Unlike conventional PCR, which only provides endpoint results, i.e., presence or absence of DNA, qPCR tracks the amplification process cycle by cycle, using fluorescent dyes or sequence-specific probes. As DNA is synthesized, fluorescence increases, and this signal is captured and quantified by the machine, giving insight into the initial quantity of DNA present in the sample. The core PCR steps, denaturation, annealing, and extension, remain unchanged. Fluorescence is measured during each cycle, usually during or after extension. The more DNA produced, the greater the fluorescence, allowing scientists to detect and quantify nucleic acids precisely. There are two primary detection methods. SYBR green, a dye that binds to all double-stranded DNA. It is economical and easy to use but less specific, as it cannot distinguish between target and non-target products. TACMAN probes. These are labeled target-specific probes. When bound and cleaved by the Taq polymerase during extension, they emit a fluorescent signal. This method is highly specific and preferred for diagnostics. qPCR is vital in gene expression studies, viral load quantification, pathogen detection, and cancer research. A key metric used in qPCR is the CT value, cycle threshold, which indicates the cycle at which fluorescence surpasses background levels. A lower CT suggests a higher amount of starting material, important in interpreting disease severity or infection levels, e.g., SARS-CoV-2 viral loads. For NET, remember, qPCR provides quantitative results. Fluorescent detection is central to real-time monitoring. Taq polymerase and probes are key players. Alright class, now let's talk about the machine that makes PCR possible, the thermal cycler, also known as the PCR machine. Think of it like the oven in a kitchen, but way more precise. Its job is to heat and cool your PCR tubes repeatedly and accurately, following the exact temperature changes needed for each PCR step: denaturation, annealing, and extension. At the center is a metal block, usually aluminum, that holds the PCR tubes. Beneath that is a Peltier element, which can both heat and cool quickly. There's also a heated lid to stop condensation inside the tubes, super important to keep the reaction going smoothly. You program the thermal cycler using a simple interface, sometimes even a touch screen. You enter the temperature and time for each step. A typical cycle looks like this. Initial denaturation at 95 degrees Celsius for a few minutes, then about 30 cycles of denaturation at 95 degrees Celsius. Annealing around 55 degrees Celsius. Extension at 72 degrees Celsius. Even a tiny change, just a degree or two, can mess things up, so accuracy is everything. Modern machines come with cool features, like gradient blocks to test different annealing temps in one run, real-time monitoring in qPCR setups, and even remote access via apps or cloud platforms. Handy in busy labs. And for your NET prep, remember, the thermal cycler is what automates PCR. Temperature changes are programmed, not manual. It's the heart of PCR in every lab. So while it might not get the spotlight like DNA or Taq polymerase, the thermal cycler is the real workhorse behind the scenes. PCR's impact is not just theoretical, it has made tangible changes in science and society. Let's walk through a few case studies. COVID-19 diagnostics. In 2020, RT-PCR became the global standard for detecting the SARS-CoV-2 virus. Nasopharyngeal swabs were collected, RNA was extracted, reverse transcribed to cDNA, and PCR was used to detect viral genes. The test had high sensitivity and became a public health cornerstone. Forensics, the DNA fingerprint. In a 1980s criminal case in the UK, PCR-based DNA evidence helped exonerate a falsely accused individual and catch the true perpetrator. The ability to amplify STR regions was central to building a DNA profile from minuscule evidence. Prenatal diagnosis. PCR allows detection of genetic disorders like thalassemia or cystic fibrosis in unborn babies by analyzing fetal DNA from amniotic fluid or maternal blood. Gene cloning. Researchers frequently amplify genes using PCR for insertion into plasmids and expression in host cells. For example, amplifying the insulin gene for cloning into E. coli to produce recombinant human insulin. Environmental PCR. Scientists monitor water and soil samples using PCR to track microbial populations, detect pathogens, or even analyze ancient DNA from fossils and sediments. Each of these examples shows how PCR isn't just a lab technique, it's a transformative tool that influences lives, justice, medicine, and discovery. Let's review the key takeaways from today's session on PCR. PCR, or polymerase chain reaction, is a method to amplify DNA in vitro, invented by Kary Mullis in 1983. It requires template DNA, primers, Taq polymerase DNTPs, and a buffer with magnesium ions. The process involves denaturation, annealing, and extension, repeated in cycles using a thermal cycler. PCR can amplify even trace amounts of DNA, with each cycle doubling the target DNA amount, leading to exponential amplification. There are multiple types of PCR: RT-PCR, qPCR, nested, and multiplex, each with distinct applications. PCR has revolutionized diagnostics, forensics, cloning, gene expression analysis, and even environmental biology. Limitations include sensitivity to contamination, primer design issues, and the lack of proofreading in Taq polymerase. For NEET and entrance exams, memorize the steps, temperatures, key enzymes, and applications of each PCR type. Understanding PCR sets the foundation for mastering molecular biology and genetics. It's a tool you'll encounter throughout your academic and professional career. Thank you for your attention. Hit the like button and subscribe for more videos.